Okay. Not very big, is it? Oh, come on. Okay, finally. What a pain in the ass. This is I sent out an email about you guys should have watched the next couple of slides on YouTube from the other class, but I'll go over it quickly. So there's one of the predictions from evolutionary theory in terms of ancestor-descendant relationships is that there should be bad design, because there's something called the panda principle, where evolution only has what is there to work with. And so a lot of times you get kind of crazy stuff like, why are humans prone to choking? And so it's mainly because our ancestors were fish, and they had a swim bladder, and some of those species that developed into kind of like a proto-lung. So there's something called a lungfish today. Eventually, the esophagus and the trachea, in terms of where humans are, it crossed, which is not a very good design. You really, if an engineer was doing this, you'd have two separate pipes, basically. Instead, we have something, there's an opening for both, and there's a valve. And so when we are chewing ravenously on a nice big chunk of ribeye, you get distracted, and all of a sudden it goes down the wrong pipe, and you choke. And hopefully someone there knows how to do the Heimlich maneuver. Otherwise, you're a goner. So basically, there's a whole bunch of instances of bad design, like the prostate gland in men. Men usually have problems later on in life. The prostate enlarges. It surrounds the urethra, so they have difficulty urinating, right? So there's a bunch of things like that. And then there's a whole bunch of different transitional taxa that you would expect in terms of, let's say, in the fossil record, or even living today. So one of the best known ones, there's a couple of them. First of all, there's feathered dinosaurs and a whole bunch of other bird-like taxa. Now, this dinosaur there could not fly. Okay? You use the feathers for insulation, things like that, and display. And then there's Archaeopteryx, which is considered sometimes the first bird. But it has a bunch of traits that are ancestral, like it's got teeth, it's got a tail, a number of other features. And then there's this guy, who's an ancestor of all tetrapods. And there's a bunch of fish-like traits in these animals, and also newly derived traits. So you've got the beginnings here of limbs, but there's still evidence of like a flipper and a tail and all that. And there's a whole bunch of other intermediate fossils as well in terms of the evolution of tetrapods. In other words, land animals with 4 limbs. And then, why do whales have lungs instead of and hip bones instead of gills? Well, they descended from a tetrapod that was living in an aquatic, semi-aquatic environment, and over time there was selection so that they lost their, in fact, they lost their legs and they turned into flippers, and so you can still see vestigial traits there. So here we have all these different fossils that we found, going all the way back to a 4-legged ancestor, and then ending up with modern-day porpoises and whales. Now, in terms of phylogenies, I've already mentioned that we usually, yes?
So, a transitional taxa would be anything that went through a microevolutionary change from common ancestors?
Yeah, well, not necessarily, it's actually macroevolutionary change.
Okay.
So usually we're going to see it in, we'll also see it in microevolutionary change too, but the thing is fossils, those things don't fossilize. So we've only got, but usually it's at the species or above level, and we'll see instances where it's kind of intermediate between one kind of taxa and another. Like you wouldn't expect all of a sudden to have a creature where the ancestor was walking around on 4 legs and all of a sudden it's got flippers.
Oh, so it's the what do you call it? Record?
Yeah, the fossil
Trail?
record.
Oh, okay.
And you're going to see, over time, you're going to see these different phases where it finally end up with modern-day whales and porpoises. They didn't all happen at once. It was a very long process.
It's basically showing like the actual, like the
Fossil record, right.
of what happened.
Exactly.
Okay.
Now, we don't always have that because the fossil record is really spotty. Okay? It's very hard to actually create fossils. Probably, I'd say 99.9% of all taxa, whether it's plants or animals or whatever, leaves no fossils. Okay? So it takes just the right kind of situation where sedimentary rock, usually a lot of ocean organisms, because they have hard shells, that sort of thing. Good question. So, here we have a phylogeny, and this is actually called a polytomy, where you have more than 2 taxa that split off. And so we don't really know much about this phylogeny, except that a platypus and a kangaroo and a beaver, they're all mammals of 3 different kinds. And then there's a turtle. Okay? The thing is, all of them have a vertebral column. Now, this taxa right here, from this on, there's a common ancestor that actually had hair and mammary glands. It was an ancestor that was probably, well, more like a reptile, except that it had these features, and that lineage eventually evolved into mammals. Okay? So we've got this ancestor. We know that turtles don't have that. They don't have mammary glands and hair. And then we've got 3 more. We've got another polytomy here. And we've got a platypus, which is an egg-laying mammal, a kangaroo, which is a marsupial, and a beaver, which is a placental mammal like ourselves. Okay? So normally what we would do is we'd say there's 3 changes here. But these are all the actual changes that you would actually see. So these mammals include a prototheria, which is egg-laying mammals. Okay. And then there's therian mammals, including marsupials that have a pouch. So the fetus gestates inside the womb, but only for a short time. Then it climbs out and into the pouch. And then eutheria beavers. Okay, and we can see that, let's see, we've got placenta. So this is placenta. Here we've got a pouch and live-bearing young. And then here we've got egg-laying, which is actually the ancestral trait. Oh, that's hair and mammary glands. So the ancestral trait is here. All our ancestors were previously to that were egg-laying reptiles. So what you do is you map onto an evolutionary tree. This is obviously, you can go from older to younger, more recent. And we can see how things actually evolved. And then here's a dinosaur phylogeny. All of the dinosaurs are extinct except for birds. And we'll get into why we know that birds are actually dinosaurs later. And then here's a phylogenetic tree of life. There's a common ancestor for all. There's bacteria. There's something called archaea, which are also single-celled organisms without a nucleus. And then out of that group, one lineage led to eukarya, and that includes us. That includes cells that actually have a nucleus and organelles and things like that. Okay? And we know this because we've taken RNA. Now, it turns out all life uses RNA. But there's been changes over time. And we can actually track those changes if we sequence them in terms of their DNA sequence. And RNA is used for messenger RNA, so you're basically trying to translate DNA into an actual protein. Okay. And actually, eukarya are more closely related to archaea than they are to bacteria. Okay. Now, there's this old human classification that's really no longer used. It's the old kind of Linnaean classification. So here are the different taxonomic levels, including the domain, bacteria, archaea. Then you have a kingdom, like animal kingdom or plant kingdom, phylum below that, class, order, family, genus, and species. If you look at humans, so we're eukarya, we're an animal, we're in the phylum Chordata, we're in the class Mammalia, we're in the order Primates, the Hominidae, and then the genus and species Homo sapiens. Okay? So you would say that, let's say a kingdom would have lots and lots and lots and lots of species for the most part, okay? Whereas if you go down to like a genus level, usually a genus level has only a few species. Now, normally what we do at this point is we actually use phylogenies. But but this is an old system that was kind of repurposed for our present use. So normally when we talk about like a species, we mention the genus Homo and the species Sapiens. Okay? So this is the old classification system. Now, up until this point, I've only been talking about one force of evolution, and that's natural selection. It turns out there are other forces that result in changes from one generation to the next in traits or alleles. The first one is genetic mutation. Okay? Now, mutations don't happen very often, and most of them either have no effect or a bad effect. So here we have DNA replication. It's not perfect. There's random errors at certain points. Random mutations are the source of new alleles, but if there's a big part of the genome that does not code for proteins. It's part of regulatory stuff, and so you can have a change there that won't have very much effect. But if it does occur within a locus that codes for a protein, it's usually going to make that protein non-functional or not functioning properly. So there can be selection against it. Only rarely do we have mutations that actually result in beneficial mutations. Okay? But anyway, this is the actual ultimate source. Now, okay. This is a reading frame for, we'll say, DNA. And it turns out proteins are made of amino acids, and each amino acid is coded with 3 letters of the genetic code. We haven't gone over the genetic code yet. There's 4 bases, C, T, G, and A, okay, that make up the genetic code. And you need 3 of those to actually get an amino acid that then is added to a chain of amino acids, and you end up with a protein. Well, these dad pet the fat dog, the fat cat ate the big rat, the guys saw the ewe and the doe. Okay? Now, these are not DNA letters, are they? But just to get you an idea of what can happen in terms of mutation. So this is the reading frame, but if you have a mutation where there's a deletion, for example, now we've deleted D, we've got a reading frame that gives us gibberish. Okay? In other words, you have a mutation of 1 letter, deletion, and you've got gibberish there. That means you're going to have strong selection against this, right? You're going to have a protein that doesn't work. And if you have, let's say, 2-letter deletion, same thing. It's all garbage. Now, remember, each amino acid is 3 letters long, so if you end up with a 3-letter deletion, let's say you have an R right there, everything else is fine, and then there's 1-letter deletion there, you'll have Dad pet the fat dog, the fat cat ate the big rat. Let's see, actually go. The big at, and then from then on it's all garbage. Okay? So in other words, we've dropped an R, and instead of having rat, we have ATT, HEG. None of these are actually words, right? They would not code for anything. Now, if you did the same thing again and you lost 3 IGR, in other words, big rat, you would see how now it still makes sense, but it's been changed. So you've got dad pet the fat dog, the fat cat ate the bat, the guy saw the ewe and the doe. So there's been a change there in terms of an amino acid. Now, whether that protein would still work or not remains to be seen, but this could be, let's say, a beneficial mutation. Okay? So, yes?
So like when we were talking in the beginning, how like bacteria created mutation and was resistant for its
Uh-huh.
So that's why that happens.
Exactly.
And with the snails.
Yes.
Oh.
Yeah. So, because there's all, with eukaryotes, there's a whole bunch of ways that the DNA actually, there's a change that it repairs itself. But it's still not perfect. So there's always going to be mutations. Most mutations happen actually in our autosomal cells that are not part of the germ line, so they're not passed down. So if you look at cancer, you get a bunch of mutations there. But in this case, we're talking about germline cells that lead to sperm and eggs. They're passed on from one generation to the next. And this is where you actually get mutations that will be passed down. And if there's a change, let's say that's beneficial, then individuals who inherit it will have, let's say, an advantage over those that don't. Okay? Okay. Now, there is another force. So mutation by itself, by the way, happens so slowly that it almost has very, very little effect on evolution. It's very, very, very slow. You actually need mutation coupled with, let's say, natural selection before you actually get something where it's happening. You've got mutations that are then kind of parsed apart. Those that are beneficial are passed on, those that are not are gone. Okay? Now, the thing is, these alleles, I mentioned that natural selection leads to changes that are adaptive. In other words, they increase those who have them, have a benefit, and they're more likely to survive and or reproduce than others that don't, let's say, have that. But there's also a random component to how alleles can actually change. And I'll give you an example. Now, we could say, let's say I'm walking along and there's a butterfly and I don't notice where I'm walking and I step on it. That's not a source of mortality that's common to butterflies. A more common source, let's say, is a bird coming along and eating one of them or something like that. Okay? Yeah, that's something where selection can actually work. But a random event like me stepping on a butterfly or a pine cone coming down and hitting me on the head, that's going to, you've lost an individual with alleles, but it's really a random event. So genetic drift is completely random with respect to whether a change is good or bad. Okay? Now, when I talked to you before, I mentioned that humans are diploid. That means we have two copies of DNA. So we have chromosomes that are copies of each other. One's from mom, one's from dad. Each chromosome has the same genes or loci on them, but there might be differences in terms of whether one locus has an allele from mom that's different from the allele from dad. So remember when I talked about genotypes, I said there were usually, if you have two different alleles, you have three genotypes. You have, let's say, AA. you have then BB, or you have AB. Okay? So those are the three genotypes. In AB, what you've got is you've got different alleles. Let's say Dad gave you A and Mom gave you B. Okay. Now, the chance of getting that allele are 50-50. Because it turns out, in terms of gametes like eggs and sperm only have one chromosome pair. Okay, one chromosome of the pair, not two. So there's a chance of getting, let's say, allele A from Mom. Might be 0 because she's actually BB. Or it could be that Mom is AB, and it turns out if you flip a coin, you get an A, and if you flip a coin the other way, like a tail, you get a B. Okay? So if you look at probabilities of A1 and A2, let's say group 1, we'll say a person, flips a coin 10 times, they get 6 heads. In group 2, the first person flips it 10 times and they get 2 heads and 8 tails. And so on down the line. Now, in the end, we end up with group 1. If we added those all up, we'd have 21 versus 19. And if we actually did a test on that, statistically, we would find there'd be no difference. But if you notice, by flipping it only 10 times, you get a change of 10% difference. So let's say group 1, you flipped it 10 times, you get a 6. That's 60%. The next one is 5. Now you'd expect, with heads and tails, you'd expect 5. But you don't always get 5. You average 5 over many, many flips. In this case, for group 2, or the person number 2, he flips it and he gets 5 times. So that means there's a change when you only flip 10 times per individual of 10%. You go from 50, 5 out of 10, to 6 out of 10. Okay? Now, if you flip it 100 times, you will still get changes, but the chances are you're going to be pretty close to 50%. And so the only real change is going to be, you can have a 1% or 2% change versus a 50 or 60% change. And then the same thing is going on with 1,000 flips, where you get even better. So 534, so 1 out of 1,000, you're going to have a change. So in the end, you'll end up with very close to 500, but it might be 491. Okay? Whereas with 10, you would only end up at either a 4 or a 5. Okay? Or with 100 flips, you either end up with 49 or 50. Okay? So the more flips you have, the more likely it is that you're going to average 0.5. In other words, half the time you get heads, half the time you get tails. Okay. Now, if you draw these colored beads out of a pot, we'll say this is the population of alleles. We talked about that earlier, the population of alleles. And we've got 2 alleles. We've got green alleles and brown alleles. And you go into a pot, you pick one out. So you draw 4 of these alleles. So let's see. Okay. Oh, you draw 6 and 4. So you have 6 brown and 4 green. Okay, you just do it 10 times. So now for this new second generation, you now have 60-40. Okay, you start out with 60-40 instead of 50-50. All right, so here's 50-50, here's 60-40. And then you, again, draw And this time you get 7 heads, or 7 brown, and 3 green. So now instead of, the expectation is 5 out of 10, but the second generation is already 6 out of 4, so it's 60%, 40% starting. And when you draw again, it's like drawing, it's just by chance, and you end up with 7 out of 3. Okay? And then you draw out of 7 of 3 again, but this time you end up, by chance, getting 60-40 again. So after the 4th generation, it's drifted from 50-50 to 60-40 to 70-30, and then back to 60-40. That's called sampling error, and that has to do with which allele on which chromosome you get from your mother or your father. Your father and your mother, they both have 2 chromosomes that they can donate. They got those from their mother and father, your grandparents, right? And it's just a matter of chance which one you actually get that you inherit. So this is what's called genetic drift. Genetic drift, so you can have changes over generations. In this case, the frequency of green or brown allele, just by chance alone, that has nothing to do with whether the alleles are actually beneficial or have no effect or are detrimental. They just drift because it's all a game of chance in terms of what you inherit from your parents. So, with that, you end up with a population. Remember, the population on a definition for a population evolving is that there is a change in allele frequencies over generations. That's all it is. In this case, it's not adaptive. It's random. Now, when you have a lot of different alleles and a large population, it's like instead of flipping 10 times, let's say you flip 1,000,000 times. So you're going to be closer and closer to what the actual population frequency is for a certain allele. Now, so to give you an example, here we have the generation along the bottom. We actually have a population size here, and we're looking at a bunch of different populations. Okay? So each one of these, like this one, we'll say, that's one population. That's another population, and then there's all these other populations here. We've got a whole bunch of different populations. But if each population only has 4 individuals, the chances of getting one, like let's say you flip a coin 4 times, you have a much higher chance of, well, getting, let's say 3 out of 4 heads, or even 4 out of 4, than you would if you flipped it 10 times. So you got a lot of variation. So with a small population of 4, you get all this variation. In this case, this allele, A1, if it's the red one, red population, it means it's gone to fixation in the population. Allele A2 has gone extinct. For the blue population, it's the opposite. Just by random chance alone, over a number of generations, in this case 20 or 19, this population has lost the A1 allele and is now all A2. So if you see on the left side, it says frequency of A1. Okay? And A1 and A2, if there's only 2 alleles, must add up to 1. Okay, now if you then do a population of 400, instead of losing or gaining, fixation or losing allele through extinction, you end up with what you would expect. If you start out at 50-50, you'd expect the population in terms of the frequency of the alleles over generations to fluctuate, but not that much. Okay? If you did it 1,000,000 times, let's see, you would probably have a population that fluctuated still a little bit, but even less. Okay? This is all from drift. So, genetic drift is random fluctuations in the allele frequencies from chance events in a finite population. It turns out that drift is strongest, like these random events from which allele you get, is strongest in small populations. So if you have a very small population, like population size 4, you get a lot of drift, very strong. If you have a larger population, let's say of 400, you get much less drift. If you have 1,000,000, you get even much less than that. There's still going to be a little bit. The only time you would get a straight line across with no fluctuation is if it was an infinitely large population, which doesn't exist in nature. So drift is still happening even in a population of 1,000,000, but it has much, much less effect on the allele frequencies of the population. Now, drift reduces genetic variation due to chance extinction. In other words, you get an A from your, let's say, one parent, and a B from the other, and you have children and you only pass on the A, and that's the only grandchildren that are available, is one of them, and so you've lost the A allele, but you have the B, or rather you lost the B allele, but you have the A still in the population. Okay? So genetic variation has now been lost. You've now lost allele B instead of having an allele A and allele B in a population. Genetic drift does not have any effect in terms of fit, in terms of what the organism is and how it fits well to the environment. In other words, does it have an adaptation that allows it to do well in the environment, or a bad mutation that gets rid of it? No. It's just strictly a random thing. It could be good, it could be bad, it could have no effect whatsoever. Now, to give you an example of what drift can do, there's something called a genetic bottleneck. And that means you have a small population. So here's an example. We've got 3 different alleles, or 3 different genotypes, 2 alleles. We have A1, which is red, A2, which is blue, and then the genotypes, we also have a heterozygote that's A1, A2, and then if they're A1, A1, you are red. If you're A2, A2, you're blue, and if you're A1, A2, you're purple. Okay? So we've got these 2 different alleles in here. Now, let's say we have and each of these is a diploid. So the red one is A1, A1. This is, A1, A1. This is A2, A2. And then these are A1, A2. Okay? Let's say you have a population of this size and a disease comes along and kills all of them but 4. Now, you've now got a population of 4, and if, let's say, you had, well, we'd have to count them up, but so many purple, fewer red, fewer blue, but you end up with only individuals that either have, are A2, and you have one that's A1, That's what the remaining population has experienced. So now we've got this very small population. When these guys breed, there's a good chance that there's a total of 6 A2 alleles, 7 A2 alleles in this population. There's 2 alleles for each individual. So these are A2, 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 that's 6. And this one is A1, A2, so that's 7. Okay. That means there's only 1 red allele in this population now. Okay? There's a good chance that that allele will be lost in the next generation, because you now have 1 out of 7 chances of actually an individual inheriting that 1 allele. You've got a 50-50 chance, but you've got 4 individuals total, 7 alleles, and 1 out of 7 is a very small number. Okay? So this bottleneck reduces the genetic variation in the population. Now, if this had happened in a very large population, and you ended up with a smaller population, but it was still large, it wouldn't have that much of an effect. Okay? But this is a small population where drift is strong. So in this case, genetic bottlenecks have greatly reduced the genetic variation. In this case, northern elephant seals and cheetahs. We know that if we sequence cheetahs and elephant seals, and they have less alleles, less genetic variation than one would expect. So it looks like sometime in the past, the population went through a genetic bottleneck, like we just saw, where it went from large to very small, and then grew again. But that starting population that then grew had already lost a lot of the genetic variation. So this is all genetic drift. The same thing can happen with something called the founder effect. In fact, the founder effect is basically a case of a bottleneck. In this case, it's not because something has wiped out the population. It's because, let's say you have a large population on a mainland that are birds, and 4 of them fly off to an island and start a new population. So you go from a large population of, let's say, 1,000 birds, and then on this island, we've now got 4 individuals that's only a population size of 4. So this is called a founder effect. And founder effects can have large effects on populations. We have the same sort of thing, but in this case, all these being wiped out, these individuals instead just flew from this big population across the ocean to this island and started a new population. And the immigrants, there were 3 of them. There were 2 of them had A1, and then 1 of them had A1, A2. Okay. Now, to see what can happen with a founder effect, there's this island in the South Atlantic called Tristan da Cunha, and it was a British possession, and 15 colonists went there in 1814. Okay, that's a small population. It turns out one of those individuals was carrying an allele for a disease that causes blindness called retinitis pigmentosa. It's an inherited disorder. If you get one allele you're fine. But if you were to get one from your mom and one from your dad, you'd have a recessive deleterious allele, and that individual would be blind. Okay? Well, because we started out, 1 of 15 actually had this retinitis pigmentosa allele, didn't show any signs, but he was a carrier. And when that individual bred with another member, that allele was passed on. There's a 50-50 chance that that allele was passed on, because most everybody else had A1. Okay? So over time, this is an autosomal recessive, because this is a population where you had an allele a frequency of 1 out of 15, or 1 out of 30 actually. That's a much higher incidence than, let's say, 1 out of 400 or 500, right? So the chances eventually over many generations of finally, way back, let's say your grandparents, one had one of those alleles, the other one had the other allele. It's passed on through both lines to one parent, and then there's this other parent. They each have one of those alleles. If those two parents breed, they're A1, A2, and they both end up with A2, that's a retinitis pigmentosa allele, it means their child will be blind. So there's a much higher, now normally, since it's a bad allele, that in a large population, the chances of inheriting 2 copies of that allele would be very small. But in a small insular population like this, the chances are much higher. And so in this case, you've got a whole population now where the incidence of carrying the retinitis pigmentosa allele are very high, and somewhat less, the chances of actually an individual having both copies are still much higher than you normally would find. And so this population has kind of an endemic retinitis pigmentosa allele in terms of offspring. Now you think, well, these blind individuals, they have lower fitness, but the thing is, if you only inherit 1 copy, it doesn't affect your fitness. And so that allele is going to continue to be passed on through time. Okay, so this is founder effect is an example, again, of genetic drift. Remember, drift is very strong in small populations, much, much weaker in large populations. Okay, now, there's mutation, there's genetic drift, and then there's something called non-random mating. If the chances of mating were random, I'd pick, let's say, I pick you right there, and I pick you right there. That's a random event, okay? And then I pick someone else. That's random, where anybody in here could breed with any other person of the opposite sex. Okay? That's random mating. But oftentimes we don't see random mating. Sometimes we see associative mating or sortative mating where certain individuals are, let's say, more attracted to other individuals. For example, let's say you have a tall woman, and she's probably looking for a man who's taller than she is. In our ancient past, it was good to have a big, strong, tall man because there was going to be some other Neanderthal out there who might want to take your wife away. And if you could defend her, that was a good thing. Okay, so this tall woman picks a tall man, not someone shorter than herself. So eventually, you have individuals tall, pick tall, let's say medium, pick medium, or in this case, different colors. We've got purple up on top, brown to the right of it, green, blue, chartreuse, brown, okay? That's assortative mating. Now, you could have disassortative mating where you have a bunch of people who like novel mates. In other words, you don't want to just have a mate from the same culture as you. You want someone different. In this case, you'd have, let's say, this guy actually mating with this guy. That's a disassortative mating. Okay? It's novel. It's like, oh, that's pretty cool. I think I like that. And you have this one mating with one of this one. Okay? Now, non-random mating can lead to changes in allele frequencies because each of these different phenotypes has certain genes and not others. Okay? What you can do is, if you have disassortative mating or assortative mating, where like is like, you can lose alleles over time, just like with drift. So, as you probably know from history, many populations, they have a government with, let's say there's a king or an emperor and an empress. Okay? Now, the royal lineages, whether you're in terms of the pharaohs of Egypt or the royal families of Europe, they felt like royal blood. In other words, being the king was passed on from parents to offspring. Now, so you didn't want your son, let's say, your prince, to marry any old common girl. You wanted her to marry into a royal bloodline, usually another family that was also powerful and that they could form an alliance. So in terms of King Tut, it turns out they've recovered the DNA from King Tut. Now, they know from history, from writings and hieroglyphs, that King Tut's parents were actually a brother and a sister. Okay? Called consanguineous mating, where you take closely related individuals and have them breed. Unfortunately, this is like having a small population, and the chances of inheriting two alleles that are actually inherited from a common ancestor are much higher. Well, over many, many generations, King Tut's parents were brother or sister. Well, he ended up with a club foot. He ended up with necrotic foot bones. He had a cleft palate. We know this from his mummy that he had multiple malarial infections. In other words, his immune system didn't work very well. And when he bred with another close relative, they produced two stillborn fetuses. Okay? In Europe, if you actually look at the royal bloodlines, there was something called the royal disease, which was hemophilia, where it was a trait that was on the X chromosome, and only those, the only people who actually had it were boys, because they got one X from their mother, and then they got a Y, and because of that, there was a bad X, and they ended up with an allele that caused hemophilia. So many individuals in the royal bloodline in Europe inherited hemophilia, or were carriers of that allele. There were also a whole bunch of others. In fact, if you actually look at the offspring that royal houses produced, over time, there was real strong incidence of inbreeding depression where you had all these children that were born with something wrong with them, or that were born stillborn, or miscarriages, or things like that. So you might have, oh God, there was, like let's say you have the King of Spain, and his wife was from the, a princess from France, and they were closely related to each other, and they bred. You had inbreeding depression, and the chances that they both had, let's say, deleterious allele, and a recessive deleterious allele, we get 2 copies, is very high. So this is what you see a lot in populations where there's kind of really strong inbreeding depression. Okay? Over time, like by the time the royal period ends in Europe, where there were no more, there's still kings and queens, but they have no power. And they don't interbreed like they used to. But by that time, there were so many bad alleles in that small population of royal blood that they might have ended up with 1 kid out of 10 that actually made it to adulthood and wasn't crazy or something like that. Okay. Now, finally, there's one other way that you can have changes in allele frequencies, and that is you have migration or dispersal. Dispersal can either be individuals or it could be gametes. So, example, this is a bat that is feeding on nectar from that flower, and it's a pollinator. It picks up pollen from that flower, takes it to another saguaro cactus, and pollinates another flower. Okay? Here's more pollen. This is gametes from coral. These are seeds, the same sort of thing. And then individuals, right? Birds. Migration is dispersal from one population to another. So you have these 2, let's say, populations that are either A1A1 or A2A2. Okay? And they disperse between them, and you have a mixing of these alleles. Right? So the A2s go over to the A1s, and the A1s go over to the A2s, and they interbreed. The populations, the frequency of those alleles changes because of migration. Now, when there's movement of alleles from one population to another, that means there's gene flow. In other words, alleles being transferred from one individual, one population to another population. It's called gene flow. When you have gene flow, the populations become more like each other over time. So with continued interbreeding here, eventually you'd end up with a population that has a pretty even mix of A1 and A2 alleles, and there'd be heterozygotes as well. Okay. Now, if you interrupt gene flow, that means you have one population that's, let's say, split into 2. That means those populations are no longer exchanging alleles. So, for example, there's these squirrels. This is the Grand Canyon. I don't know if anybody has actually been there, but that's the Colorado River. Now, up on the mesas, on this side, the north side, there's a tassel-eared squirrel, and on the south side is a close relative. Before 7 million years ago is when the Colorado River started flowing through this area and cut down into the canyon and split these populations from the north side and south side. At one time, they were one population of squirrels, okay? There was no canyon here. It was all big mesa top. But over time, those populations eventually got separated by the Colorado River. It turns out squirrels can't fly unless a hawk comes along and grabs one and carries it over to the other side. And then it's probably not going to survive and reproduce. Okay? So, it turns out that in terms of gene flow and migration, if you have a population that is separated into two smaller populations, and there's no gene flow going on, eventually over time, those populations will change, either from selection or from drift and mutation, so that eventually there'll be two separate species. So if you restrict gene flow, that means there's less migration, right? Then there's greater population differentiation from the forces of evolution. Forces are natural selection, mutation, genetic drift, random mating, and migration. Okay? And over time, that'll result in perhaps speciation. So now we have these two squirrel populations. They're actually different species now. Okay. So here's a question. In the face of environmental change, let's say new selective pressures, which is more likely to go extinct? A species with a large gene pool or a species with a small gene pool? Now remember, anybody? Small, right, because of genetic drift. So, if you have a large gene pool, you'll have drift, but it won't be much, and you're likely to not lose your genetic variation and go into a situation where you have a population with no genetic variation, and they're not able to respond. Now, why are they more likely to go extinct? There are fewer alleles than natural selection can act on, that's true. Genetic drift is also true, has a larger effect. And then it turns out the last one, both A and B are true. It's true. So species are more likely to evolve rapidly by natural selection if they have a small gene pool or a large gene pool. Natural selection now, not drift. Large, actually. So drift can overcome natural selection in a small population. But in a large population, where many different alleles are in the population, selection has a lot of variation to act on. And the chances of losing one allele or another just by chance is very small. Okay? Okay. Now, this is Darwin's mystery of mysteries, and that is how are new species actually formed. And there's a number of different mechanisms we're going to go through. So remember, species come from a common ancestor. And then we're going to look at mechanisms of speciation. You can see what happens. These are Hawaiian honey creepers. So probably 5 million years ago or something, the ancestor of this group of birds managed to make it to Hawaii from the mainland of Asia. Okay? It was by luck. Most birds that, let's say, got blown off course by, let's say, a hurricane or something, would end up out at sea and dead. But they managed to land on an island in Hawaii. Looks like it was Kauai. And there were no birds on this on the Hawaiian Islands back then. And so these birds spread and bred, and then there was new islands, and some flew over to the new islands, and then some flew over to even newer islands. And there was a lot of different kinds of habitat, and you ended up with, something like 53 different species of these Hawaiian honeycreepers. Then the Polynesians showed up about 1,300 years ago, and these things started going extinct, and then finally when Europeans showed up, now there's not very many left. But this is an example of adaptive radiation, where you had two individuals at least from one species. One was male, one was female. They managed to get in the same place. They managed to breed. They started a population. There was lots of food. There were no predators on the Hawaiian Islands that could eat birds then. And over time, some populations got isolated and they speciated. Mainly because of natural selection, so that's why you have all these different beaks. Some beaks are good for drinking nectar, like this individual and this individual. Some are good for cracking seeds, like that. Some are kind of, general purpose sort of beaks. Maybe for insects, something like that. So you've got all this speciation happening on different kinds of diet. Now, there's also sexual selection going on, so you can see that with butterflies. And then another good adaptive radiation is this anole lizards in the Caribbean, where you see different colors and different habitat types. But each island has similar species or similar phenotypes, but they're not related. Okay, so it's a process of forming new species. Now the question is, what is a species? And this has been something that has dogged biologists for a long time. We think we know what a species is, and that is a group, a population that can interbreed among themselves but not breed among other species. Okay? So we've got giraffes. We've got elephants. The thing is, a lot of times you can have hybrids, and that would kind of violate what the definition of a species was. So, species is the smallest evolutionarily independent unit. It implies mutation, selection, gene flow, and drift operate on populations separately. Species consist of interbreeding populations that evolve independently of other populations. The essence of speciation is lack of gene flow, like those squirrels. They then, species, because they're only breeding within that population, let's say there's another population that's closer related, they're not exchanging alleles anymore, and there's a boundary for the spread of those alleles. Now the thing is, there's a big debate, are species real like H2O is real? H2O we know has 2 hydrogens and 1 oxygen. We can say it's actually real. There's also categories that are kind of arbitrary, like what religion you are, okay? Now we can say religion seems to be somewhat real, but there's a lot of things that maybe there's different sects of the same religion, okay? Species are kind of like that because some populations have not completely separated yet and formed new species. So we'll leave it there. Now I'm going to open up some Top Hat questions for the stuff we've just covered. I'm going to close Top Hat for the questions that were already up. So if you haven't answered them now, you better do it quickly. Okay? Okay, I'm continuing on. What is a species? One of the problems is that species aren't real, like, let's say, H2O, water. We know water is made of 2 hydrogens and 1 oxygen atom. And that is exactly water. So water is not just a category, but it's an actual real thing. Species are not quite that way. They're not necessarily arbitrary categories like, let's say someone is Danish, and then that's a category, a citizen of Denmark, okay? But it's not like, let's say someone then immigrates into Denmark, and they become a citizen. Then they're a citizen of Denmark, part of that category, okay? But it's not like all Denmark people are H2O, okay? Same thing with religion, for example. So, or let's say categories of small, medium, and large, okay? Those are our arbitrary categories. Now, species are kind of like Denmark. And we use similarities and processes to delineate what are actual species and what aren't. But there's a number of different definitions. So the morphospecies concept are species are groups of individuals that are morphologically similar and clearly distinguishable from individuals of other groups. So, you use morphology, anatomy, that sort of thing to actually group species. Now there are problems. There are a lot of species that show a great deal of variation over, let's say, a wide range of geography. So you have many polymorphisms where they're actually, let's say, plants. They're the same species, but they look pretty different. There's geographic variation. There's also species that, it turns out, they seem to be the same species, but they're actually different. They're called cryptic species. Now, the species concept that is used most often is the biological species concept. And that means a species is a group of actually or potentially interbreeding organisms that can mate and produce fertile offspring. Now, the thing is, there's problems with that. First of all, you don't know in terms of, let's say, fossils. You can't fossilize whether they potentially interbreed or not. So we don't really know. For example, you might have some fossils and it might be the same species, or it might be something that looks like the same species, but you don't really know. It says, let's say you have geographically separated groups, but you put them in the same area, and it turns out they can mate. Or you have some gene flow where there's hybrids. So, gene flow, mating between 2 different groups, and it produces hybrid offspring. And then phylogenetic species, the smallest set of organisms that share an ancestor, a most recent common ancestor, can be distinguished from other such sets. Okay, so let's say we look over here. Oh yeah, one thing I do want to show. So, like for morphospecies concept, you can actually see how we can use morphology to group things. Okay, garter snakes. But there might be variation. And we use this for fossils, but we can use it also for living things as well. the biological species concept is this. And you can actually see that a barrier is established, two populations diverge genetically, but are still reproductively isolated, and then you have daughter species. You have reproductive incompatibility. So, in other words, they could breed at one point, but they get to a point where they can no longer breed anymore. And then we go down here, and we can say, in terms of the phylogenetic species concept, that perhaps all these are one species, and or if you want, you can do it in different size clades. So we know this clade includes this one and all these other ones. Okay? So the primary problem is that at what scale do you actually recognize groups that form a clade? And we could actually get down to populations and not, or subpopulations and not just populations. Okay, so species, I already talked about H2O. Now, biological species concept states that species are defined by who can successfully breed and produce viable, fertile offspring. Now, it turns out all dogs are the same species despite the considerable morphological variety. Okay, so Great Dane, little Chihuahua or something, and it's like, are you my species? I'd rather not find out. In other words, even though they look very different, they actually are the same species. Okay, so speciation is actually a three-step process. Gene flow is disrupted between populations. So with the squirrels at the Grand Canyon on the north and south rims, there used to be no canyon. It wasn't until water, one of the rivers, the Colorado River, was diverted, that it changed course 7 million years ago and went through this uplift called the Coconino Plateau. Over 7 million years, it created a canyon about a mile deep. So as before there was one population of squirrels, the canyon cut those population in two, the ones on the south side versus the ones on the north side. The plateau sloped, so the north side was about 7,500 feet, the south side was about 6,500 feet. So there's differences in elevation and therefore amount of oxygen and temperature, things like that. So that disrupted and created a barrier between the two populations that's called vicariance. And then the gene pools, in other words, those two populations became isolated from each other. Then populations accumulate differences in alleles and traits by the forces of evolution, whether it's mutation, drift, non-random mating, migration, or natural selection. Eventually, reproductive isolation evolves. It's either prezygotic isolation, in other words, before fertilization of the female's egg by sperm, or post-zygotic isolation, which would mean afterwards. This is a nice little video that kind of explains that sort of thing. It's a mountain instead that isolates the population, so please watch it. So here it is with an actual chart. here we have a population, an ancestral population, and there's perhaps all these different, each one of these is a population, but there are full population. So these are all subpopulations, and they exchange alleles because there's breeding between all of them. And then later in time, this reproductive barrier emerged. We'll say at this point it's actually a geological or geological structure, let's say a mountain or that river, something like that. And now there's one population, and now there's actually two. Population on the left, and then population on the right. Okay? There's no longer any gene flow between these populations, right? So they're isolated. They accumulate differences. They diverge. They develop reproductive isolating mechanisms. Members of populations are incapable of interbreeding. Populations are now separate species. So there's, in terms of geographical isolation, there's different modes of speciation. Now the most common form is called allopatric, which means allo, different, patric, homeland, speciation. And it means that there's geographic separation of populations through something called either vicariance, which is a geological barrier or geographic barrier that separates a population into two or more groups, or through dispersal from, let's say, a large population onto an island, and the formation of a very small population with a founder event. Okay, so here we've got one large population. This barrier starts to develop. Now you've got two smaller populations. They start to diverge, and they diverge even more. And then this barrier is removed. Okay? And now you've got these two populations that have come back into contact with each other. Okay? The thing is, they've diverged at this point. So, in this case, we can say if they can still breed together, they're still one species. But if they can't, then they are now separate species, according to the biological species concept. Now, so this is vicariant speciation. This is the same sort of thing. One population, a small group of individuals disperses to, let's say, a localized colony that is now isolated. It diverges. And then, let's say eventually, those two populations come back into contact again. Again, if they are able to breed, they are still one species. But if this new population, the small one in red, and this overlap with the yellow can no longer breed, then they are separate species. Now, forget about parapatric speciation. And then there's something called sympatric speciation. That speciation, same homeland in the same place. Okay, host plant shifts with insects, polyploidy, hybrid speciation. There's a number of different ways. Let's take a look. So here's vicariant allopathic speciation. We've got one large population. Okay, we can see that there's 5 different body colors, and they each have equal numbers, so it's 20% for each of the total population. Okay, now the river has changed and has now cut these populations off from each other. They started out with the same, let's say, proportion of dark green, light green, all that. Okay? And so, 20 generations later, the left bank shows that there are more darker lizards. The right bank, after 20 generations, there are more light-colored lizards. Plus, there is a mutation, the spotty mutation. Perhaps there's a difference in soil type. And the soil is like gravel that looks like spots. Now, 40 generations later, we see that the spotty mutation has become more common over here. And there is no spotty mutation over here. And this population is certainly darker. And this population is certainly lighter with those spotty mutations. And so we also have differences in the different color morphs in terms of the frequency or percentage. 60 generations, same thing. There's even more, and it's almost all spotty mutations on this side, and almost all darker forms on this side. Okay, so this is a real-world example. The Isthmus of Panama is that land bridge between North America and South America. So, in fact, here we see the Panama Canal. That's where the Panama, and that's where the canal is. I'm not sure exactly where. I think probably here or something like that. Anyway, about 3 million years ago, before 3 million years, all there was water. All the way across. Okay? And so these were all one population of snapping shrimp, or a number of different species of snapping shrimp, but it's like, we'll say, this and this. Before the land bridge, they were one species. The land bridge came up and separated them. This is vicariant speciation. And this separated these populations of snapping shrimp on south of the Panama, or west of the Panama Canal and east of the Panama Canal. These populations started to evolve and become distinct, but you can see that snapping shrimp species are more closely related to sister species on the other side of the isthmus than to species on the same side. Okay, so this and this are actually sister species, and they're more closely related than this is to this. Okay, so you now have different speciation because of this allopatric vicariant barrier. Okay, we'll stop it there.